Thanks, Eric, for the kind introduction, and thank you for giving me the opportunity to talk a little about Quad and what we're doing, as well as some of the challenges when it comes to user requirements. So before I start, we founded Quad Technologies focused on really addressing some key bioproduction challenges within the CAR-T and TCR T-cell immunotherapy process. And so that's what we really focus on in Quad, is building novel and differentiated tools to help address some of the key challenges in the next paradigm within oncology. So for some that might not know what CAR-T is or TCR, it's a engineered T-cell that you can actually train. We reinfuse that back into a patient, and it actually goes after different oncology indications. This has been really a huge highlight that Immunotherapetics, Novartis, Kite Pharma has been leveraging to treat a variety of different oncology indications, ranging from pediatric ALL to AML and a variety of different others. And how this process works is essentially they either do an autologous or allogeneic approach where they source patient apheresis material, purify T-cells, and then transduce those T-cells to express which is known as a chimeric antigen receptor or a TCR, which is a T-cell receptor. And those are now trained to go after different oncology indications. And so there's a lot of interesting processes within that workflow that I'm going to be discussing a little bit further. So one of the very cool things about this technology is that they've sustained and have published over 90% complete remission of certain leukemia indications. I mean, this is really the next generation on how you treat oncology today. And people have been actually looking at different methods in terms of trying to address non-Hodgkin's lymphoma, Hodgkin's lymphoma, and then there's some preclinical research that's going on with MAGE-A3 and as MUC1 looking at different sort of solid-state tumors and using these CAR and TCRs to actually treat other cancers outside of just blood-derived cancers. However, the challenge with this process, it's a pretty arduous process. You're essentially sourcing patient material. You're sourcing apheresis that is not defined, highly variable, and you really need to define that process. And so how this workflow works is essentially you take apheresis material, which is given by a donor. You typically have to purify T-cells, and then you have to transduce those T-cells with your either lentivirus or adenovirus, or through an electroporation and then doing mRNA to then express your CAR or your TCR that's specific to that oncology indication. Then you have to expand those T-cells using different expansion technologies, typically CD3 and CD28, and then that gets up to confluence so you can go into dosing and then redose that patient downstream. Now, this research has been going on for quite some time. People say, oh, well, CAR-T just started about 4 years ago, but it's actually been going on for almost 25 years. It's just right now that you're starting to get some significant ethical dosing within a lot of these patients. It's starting to create a lot of buzz. Now, with that being said, I mean, the translational research is really where it started. So that's folks at UPenn or Sloan Kettering or the NCI have been working on this for quite some time, but translating that into a biotech and a CMC process is not trivial. There's a huge gap and a huge bridge going from something that's within translational research up and all the way to the CMC. And so it's really highlighted within, call it about 3 different, facets of this bridge. So number one is that typically, classically, when you're in translational research, you're working with T75 flasks, you're working in a very open system. People have been taking proactive, methods to actually close the system down using apheresis and automation and essentially doing serum or apheresis and plasma bag transfers. And so people have been marching towards closing that system, but still it hasn't been really completely closed as of yet. In addition, you need to have GMP qualified reagents to really support a lot of these clinical trials and these GMP and these bioprocessing workflows that you really need to get to the end patient. Lastly, there's scaling challenges. When you're dealing with autologous, you're dealing with one starting population that's going to treat one patient. And so there's scaling challenges associated with autologous, but even more so with allogeneic, where you source from one donor and then you actually have to build a huge manufacturing operation to then treat multiple donors irrespective of HLA dependency. And so bridging this gap is challenging, but really even more challenging is defining the requirements on a field that's constantly changing. And so from product development, on which what Quad has been doing within the space, it's been hard to really define some of these requirements that these CAR T and TCR folks are really marching towards. And so we need to take a step back. We need to look at, okay, how do we really define these requirements and build products that can really streamline some of their research, number one. And number two, streamline a lot of their bioprocessing and really generate some great clinical outcomes downstream. So it needs, as an engineer myself, you have to start off with assumptions and you have to validate those assumptions through literature. And so what we found out and what we know is that with respect to CAR T, depending on who you ask, it's the ratios between CD4 and CD8 could influence efficacy downstream. That's a lot of the work that's done at Stan Riddell and Cameron Turtle's lab at the Fred Hutch. In addition to that, they found that more naive T-cells actually generate more persistence. So people are drawing conclusions that if you have more naive T-cells, you can actually decrease dosing and you can actually generate a better response in the clinic. The third thing is that purification is essential. Once again, as I mentioned before, is that you are starting with kind of a gamish, if you will, of cells. And a lot of people are fearful of transducing the wrong cell, whether a B cell or an NK cell. And so you really need to start with a defined population from your donor that gives you essentially the apheresis. Lastly, right now, because it's such a moving space, just like Quad, we're actually struggling to catch up because everything is changing very fast. And researchers and translational researchers are using fairly antiquated technologies that haven't really been differentiated for a while. And to that end, there's a challenge when you're doing purification T-cell activation known as debeading, which actually is a huge headache within the space, which I'll discuss on how Quad's tackling that. So how's Quad tackling these challenges? As I mentioned before, we take a step back, you have to talk to a lot of customers, you have to talk to key opinion leaders within the space to understand how the requirements interact with the downstream product and their CAR T therapy, and as well as where they're going with it. And so we took back and we looked into the literature and said, okay, let's look a little bit more granular at the process. And so Carl June, being one of the big major KOLs within the space, outlined his process very nicely. This is his NYSO TCR process, very similar to the CAR T process, and you can see it's broken down into some pretty nice steps. So the steps outlined, as I mentioned before, is that in enrichment, you have to define your population. Typically, you're enriching CD3 positive T cells. Perhaps you're enriching CD4 or CD8. Then you activate and transduce those T cells with your vector, expand them, and then you have to do a bead removal step. So this bead removal step is actually a huge hiccup within the industry right now. So what we've been identifying is that as you go through the whole process and you have to remove these magnetic beads that you actually use to purify and activate your T cells, you actually suffer from a 30% loss. And so as an engineer myself, a 30% loss downstream at the end of the process is a huge challenge for these folks. And so we said, okay, let's take the bead removal step and see how we can build a platform around that and really understand on how we can enable and decrease that loss, as well as enable purification and T-cell activation with that same platform. And so what we developed is a novel hydrogel that we call QuickGel. Now, QuickGel allows you to capture and release cells off of substrates. There are actually many technologies you can actually enrich a cell or adhere a cell specifically onto substrate, but there's actually not a lot of technologies where you can actually viably decouple a cell from a substrate without any deleterious effects on the end product. And so we took that mantra in mind. So we took that mantra in mind to actually enable the ability for facile release of cells off of substrates, whether it be polycarbonates, polystyrene, Xenor, PMMA, PDMS, magnetic beads, which is showed on the bar graph to the right, as well as develop a hydrogel that's biocompatible, has been used within FDA processes before, and to really enable some differentiated features. And so this is a fully phase change material that allows you to do that facile release of cells from substrates. So if you recall, the debeading step's the problem. So we understand what the problem was, and then we said, we have a platform to operate QuickGel, and said, well, how do we move the beads faster? So if you remember what I said before, is that facile release, you can easily release cells off of substrates. So we said, why don't we take a step back, take the existing beads on the market, coat it with our QuickGel chemistry and derivatize a capture protein to the surface to allow high throughput cell separation, except you don't have to have the 30% loss downstream because of your bead removal that's going to adhere cells at the end of the process. And so that's when we formed a product called MagCloudz that we actually use to leverage within the research use only market and then parlay that into some GMP regulated manufacturing with respect to CAR T. So how we solved bead detachment is a fairly straightforward workflow. Essentially, you take MagCloudz, derivatize with a capture ligand that's geared towards T-cells. Those T-cells, you can use MagCloudz with in combination of apheresis or PBMC, and then those T-cells would then bind externally to MagCloudz that you can magnetically now manipulate and purify those T-cells, except the added difference is at the end of the process, you can depolymerize that matrix and then have magnetically label-free cells at the end of the process. And so this is a very fast and easy process, and I can kind of paint on how fast you can actually release cells from a substrate. And so this is a little movie because I'm a huge proponent of movies because they sell, essentially say 1,000 words. So these are our MagCloudz, spherical in nature. Tiny white dots are magnetic beads, cells that are bound to the surface. And so within about 7 seconds, you'll see the plane shift, and that's adding the release buffer. You'll see that QuickGel nearly instantaneously disassociate, and you'll see those magnetic beads start to float away. And so this allows you to then clear those magnetic beads without any sort of residual carryover or losing your population downstream. So as they say, when it comes to science, the proof's in the pudding with respect to the data. Proof of concept data is great, but it's really going into designing a nice system. And so what we did is we collaborated with some KOLs at UMass Medical, and we took essentially a mock system with human umbilical cord blood that would mimic essentially lymphodepleted cells within a CAR T process. And so what we did is we took biotinylated anti-CD3 with our MagCloudz to do high throughput cell separation from a pretty complex suspension. Again, with the added feature at the end of the process, you actually depolymerize that matrix and clear those magnetic beads. So what we demonstrated is that from a starting population with umbilical cord being around 6.5%, at least in this case, where normal peripheral blood is anywhere between 40 to 70% of a given fraction, that we started off with a 6.5% positive population, but at the end of the process, you looked at 96% pure with very limited contaminant and residual carryover of magnetic beads, and also while retained very nice viability at the end of the process. And so it allows you to, again, keep high viable cells and high pure cells within the process that you don't have to have that detriment of clearing your magnetic beads downstream of your cell therapy manufacturing process. Now, as I alluded to before, ratios of 4 and 8 could play a difference, so we wanted to build the applications further and look at how do we purify CD4 and CD8 positive populations, because at the end of the day, what the literature has been saying to us is that this might actually play into an effect. So you need to actually build purification tools for CD4 and CD8. And so we looked at doing purification of CD4 and CD8, and we got some very nice results that actually active collaborators are now publishing application notes on. Typically, within a given PBMC, you get about 25% CD4. We enrich up to 96% pure without any residual magnetic beads. On the other hand, you look at CD8, where it's about 15%, and we'd be able to purify up to 96% pure at the end of the process. So once again, what we did is we took a step back and we looked at the workflow and really understand on how the workflow plays in its entirety with respect to T-cell immunotherapy and really where the pain points are at the moment. And so what we can appreciate is that, at least on the enrichment side of things, we can define that population a lot easier while actually addressing the downstream issues of magnetic bead removal and clearance. And so that's something that we developed a product initially to really tackle that challenge. So we wanted to look at it a little bit different. Because now we represent 1/3 of the process, we want to say, well, how far can we take this platform? And we wanted to explore how well we can activate T-cells. And so within CAR T manufacturing process and with CAR T therapy, the manufacturing process really essentially is a huge function of how robust a therapeutic is at the end of that process. In other words, that whatever tool that you design, you actually directly influence that drug because it's a living drug. And so it turns out that activation is a huge component of this. As I alluded to before, is that naiveness and T-cell naiveness is distinct, and that whether or not you have more naive T-cells versus more T effector cells could play into persistence. But there's a catch-22 here, right? Is because you need to grow these cells, but you also need to retain phenotype. And as you grow these cells, these T-cells become more and more differentiated over time, right? So you go from naive T-cells to T effector T-cells. And T effector T-cells, really no one within the space really wants, because they don't persist very long in vivo, and that's a huge challenge for them. So we said, okay, how can we make a system to couple our proprietary QuickGel and allow us to modulate essentially the naiveness of the T-cells and see what we can get in terms of proliferation, as well as also still addressing the magnetic bead clearance challenge that people use for T-cell activation. So first we wanted to compare and say, okay, let's look at control beads where you actually can stimulate these T-cells using control magnetic particles that have those activation markers that are derivatized on it, and then compare it to our hydrogel or QuickGel complexes, and then release the beads or do that debeading step and see what we get at the end of the process. And so within the bright field images, you can see on the far right that on the 40X, you can see there's still magnetic beads, remnants within the control beads. So that's a challenge because then they have to clear that a little bit more. But however, within the hydrogel complexes, it's fairly clean, and there's no residual magnetic bead carryover. So then we took it a step further. We want to understand, within the culture system, what sort of mix of CD4 and CD8 cells that we have. Typically within the control bead population, whatever you start with ratios between CD4 and CD8, you typically end with at the end of your culture. So there's no really influencing factors that you can actually build, essentially a better ratio between CD4 and CD8. And so we compared that to our hydrogel complex. Interestingly enough, we actually saw preferential outgrowth of the CD8 positive population that allows you to get more of a 50-50 split between your dosing downstream. So this could play into a significant part on how they do their formulation downstream, because now you have a little bit more of even keel on both cell types between the CD4 and the CD8. In addition, with respect to fold increases of the proliferation of T cells, you can see that it's pretty comparable with respect to the control beads. So we were pretty happy about that. Big difference here, as I mentioned before, is that we're getting much more CD8 growth than CD4 growth in comparison to the control beads. Now here's the interesting thing. So the interesting thing, as I mentioned before, is that T-cell phenotypes really play into ethical dosing. And so there's some interesting phenomena that we've identified that we're still working on and drawing some significant conclusions on. But we've been seeing a trend that, based off of our hydrogel, that we've been actually seeing that we have preferential expansion or retention of naive phenotypes. And so to characterize that, you have to look at certain subtypes called CD62L, CCR7, and 45RA, and we've been seeing an uptick on those markers substantially across the board in comparison to the control beads. So this is something that we're really excited about, that we're building products towards, because now we have the ability, not just within facile release, but also the tunability to actually modulate what sort of phenotypes you need to get at the end of your process and retain more naive phenotypes so you can actually generate better efficacious CAR T cells at the end of your process. So once again, what I present to you is how quad and how we address really the ebbing kind of changes in requirements that are constantly being reformulated and retaught through literature. And so what we pride ourselves is working with the KOLs within the space to really understand those user requirements and specifications. Now you can't really lock down what the true requirements are, and so that's the big challenge, but you can really address common themes within some of these requirements. And those themes could be, for instance, that we know that retained naive cells are important because they have been published pretty extensively, but we don't know what the exact quantity of those naive T cells would be after they're transduced to the CAR vector, or the exact phenotypes, whether they want T central memory cells or T stem cell sensory memory cells. Secondly, we don't understand the true ratio of CD4 and CD8. That's still ongoing within the Fred Hutch and some other folks that are following suit. And so that is yet to be optimized to really understand what the big implications there is. And then lastly, which I really didn't cover too much, most of the industry is focused around autologous, but there's a huge, also a huge focus with allogeneic, and those requirements, we don't understand if those are going to be distinct, if they're going to converge on the same requirements, or they're going to, or really where the industry is going there. And so there's some challenges with respect to addressing the allogenic and autologous-based workflows. So with that being said, I'm happy to take any questions on the panel, and I'm going to bring up James to talk a little bit more about the gene therapy. Thanks so much.